country, free country, led by the flag for which we stand. Blue for the mind that seeks to do that. Sing the day, white for the heart that's pure flame. Alma mater, peace and with success, with truth and love, with conquer all, for God land, and as God give us and great success, with truth and love, will conquer all, for God our land, and as with truth and love, let us unite be. Glorious college name. High on the hill, a beacon light is sheen. March on to glory is a message. Spread through the love of far and wide. Loyal to its guidance. Teacher and pupil side by side. Alma mater. God give us and great success. The truth and love will conquer all. For God our land and SFS. Alma mater, God give us and great success. With truth and love will conquer all. For God our land and SFS.
Am I visible? Hello?
Yes, I can see you.
Yes, Father. Hello and good morning to all. This is Dr. Susan John, Assistant Professor, Department of Physics, SFS College. We, the SFS management and faculty, are indeed happy to host this webinar on recent developments in genetics and their impact on religion and society. Let me briefly introduce to you our esteemed institution, St. Francis de Sales College, Nagpur. St. Francis de Sales College was established in 1956. It is a Christian minority institution managed by the Catholic Archdiocese of Nagpur, inspired by teachings and love of Jesus Christ, concerned with holistic developments of the students. Our college is government-aided college affiliated to Rashtra Sant Tukadoji Maharaj Nagpur University and has been accredited with A grade with CGPA 3.23 by National Assessment and Accreditation Council NAC Bangalore, an autonomous institution of the University Grant Commission UGC. CDRES, Center for Dialogue Between Religion, Education, Science and Society, is a forum launched on 24th January 2006 by the hands of late Archbishop Abraham Virthikulangara, Father Job K., Director, Indian Institute of Science and Religion, Delhi, Father Anthony D'Souza, ex-principal, SFS College, and Father Alan Tavares, to blend harmoniously and creatively the latest finding of modern science and deepest insight of religion to build up a better India in her multi-religious, multicultural and multiracial context in collaboration with Indian Institute of Science and Religion, Delhi, IISR. It has been carrying out its noble and timely mission by jointly conducting conferences, seminars, essay competitions, debate, etc. on different themes related to science-religion interfacing for the staff and students of SFS College and other educational institutions. The awareness is growing today that science-religion dialogue is an urgent need of our times and provides a platform for scientists, religious thinkers, and other scholars of the society to share their knowledge and experience. on various issues related to the relationship between modern science and contemporary religions. This time, the national webinar centers on recent developments in the field of genetic sciences and their impact on religion and society. Several reputed scholars will share their ideas on the theme. We consider it to be our privilege and honor to have you for this special event. The sequence of today's program is as follows. First, we have the inaugural speech by His Grace Most Reverend Elias Gonsalves, the Archbishop of Nagpur and the Chairman of SFS College, followed by enlightening talk on by our distinguished resource person, Father Job K., Director, Indian Institute of Science and Religion, and Dr. Susan John, Assistant Professor, SFS College. I am sure we are going to have a great session this morning. I would request you all to stay tuned till the end. You can post your queries to the concerned resource person in the YouTube. Now, I request Father Praveen D'Souza, Department of Philosophy, to please take over the proceedings of the inaugural session. Thank you, Father. Over to you.
Thank you, Dr. Susan, ma'am. And good morning, everyone.
Good morning.
I deem it my privilege to give a brief introduction about His Grace, Most Reverend Elias Gonsalves, Archbishop of Nagpur and Chairman of SFS College Society. His Grace has been taking great interest in motivating schools and our college under his care in the field of education and also in the overall development of all its stakeholders. Similarly, many social outreach programs, spiritual activities, and the like have also been made accessible digitally under his leadership in these days of the pandemic. His Grace was installed as the Metropolitan Archbishop of Nagpur in January 2019. He has been in charge of Catholic education in Western Region Catholic Bishop Council, comprising of Maharashtra, Gujarat, and Goa. His Grace was instrumental in forming Western Region Catholic Foundation for Education, WRCFE, as registered company under Companies Act 2013. His Grace is promoter of Education on Family Values online program for Western Region Catholic schools. A hearty welcome to you, dear Archbishop Ji. We are eager to hear your gracious words as you inaugurate this webinar. Over to you. Bishop Ji, kindly unmute yourself. Please unmute yourself. No, we can't hear you, Your Grace.
Yeah. Okay.
Yeah.
Sorry, I was not finding that.
Okay.
Since I have no accessibility to the computer, it is just come the electricity. I will return once again to rejoin because this was the problem here.
Okay.
A little while ago, but it is better that I continue on this now. Good morning to Father Job and Father Praveen. Good morning.
Morning, Bishop Ji.
Sorry for little inconvenience because we lost the electrical connection and therefore I have to connect to you with mobile, which is little inconvenient. But anyway, the topic today is quite interesting. And Father Job has been actively involved in this ministry of science and reason. And I'm sure all our students are lucky to have more discussion and knowledge about this subject. This is a great challenge in present day world that science and religion, this should go hand in hand. They both are complementary to each other by their equal presence and existence in today's modern world shows us how we can respect each one's identity, and at the same time we can grow together. Recent years, several works have been done on genetics, which is the topic today, a very interesting topic and a topic that which is relevant for today. On genetic engineering, there are so many things are developed with major focus on its importance, ranging from increasing plant and animal food production, improvement in medical treatment, and production of vaccines and other useful drugs. Methods in the techniques involve the selective breeding of animals and plants, hybridization, that is called reproduction between different strains of species, and recombinant deoxybonic acid. However, this development have other sides also. Dear friends, today science with the help of technology has become most powerful tool in the hands of human science. It can make or break our world. Even it can clone humans also. Common wisdom tells us that the best strategy to pursue in this situation is to find ways and means to maximize the good effects of science and minimize its ill effects. One should always keep in mind Einstein's famous quote, "Science without religion is lame, and religion without science is blind." This can be best achieved by harmoniously blending scientific might with ethical values based on sound religious practices and principles. Friends, we are in this pandemic time, and we are very well aware that how all the nations, along with the United Nations, WHO, World Health Organization, have come together to find the vaccines on COVID-19. And this effort has brought the world together in order to achieve this pandemic, achieve and overcome this pandemic. Today, as we reflect and think about the present day challenges, we need to also see how we can overcome so many diseases which are due to genetics. Diseases like diabetes, diseases like hereditary diseases, which could be overcome by simple methods of genetic science. Today, as we reflect and deliberate on this topic, I'm sure all of us will benefit and gain something very important and valuable for our knowledge as well as development of science and religion. I wish all of you, all the participants, all the resource persons, all the best for today's sessions. And I pray and I thank God for this opportunity given to you by our Father Job, who is very much into this mission. God bless you, all of you, and have a nice day. Thank you very much.
Thank you.
Over
Thank you,
to Father
Elias.
Praveen.
Yeah, thank you, Your Grace, for blessing us with your insightful and gracious words. You have rightly brought out the complementary role that science and religion play in our day-to-day lives. We are indeed grateful to you, Your Grace, for sparing your valuable time. Please do continue to guide us and pray for us in all our endeavors. Now, I hand over the proceedings to Dr. Nikat Naqvi, Associate Professor and Head of Botany Department, St. Francis Jesus College, Nagpur. Over to you, ma'am.
Thank you, Father Praveen D'Souza. Good morning, everyone. I, Dr. Nikat Naqvi, Associate Professor and Head of the Botany Department, St. Francis de Sales College, Nagpur, welcome you to the first session of National Webinar on recent developments in genetics and their impact on religion and society. I am honored to introduce the esteemed guest speaker of this session, Father Joe, Director, Indian Institute of Science and Religion, Delhi. In addition to postgraduate degree in physics, he is MA and PhD in History and Philosophy of Science from University of Maryland, USA. This itself speaks volume about his multifaceted personality and vast knowledge. He is Professor in Philosophy of Science, Cosmology in Janadeepa Vidyapith, Pune. He has been a visiting professor in many renowned colleges and universities in Chicago, Bangalore, Nagpur. He has, to his credit, 10 books. He is a keen researcher with 160 papers in national and international journals of repute. He has delivered keynote address and also presented papers in over 180 national and international conferences. He has pioneered science-religion dialogue in India. He is founder of Indian Institute of Science and Religion, Association of Science, Religion and Society, CBCI Committee for Science, Religion and Society. He is the chief editor of Omega and Indian Journal of Science and Religion. He has organized 13 international symposia in science religion interaction. He has also provided support in founding 20 science religion dialogue centers in various parts of India. He has been conferred with number of academic awards, Lifetime Achievement Award for promoting education and science religion dialogue in India, John Templeton Science Religion Course Award, author of Outstanding Academic Book of the Year Award, to name a few. He has held many administrative responsibilities as well, like Founder Secretary of CBCI CSRS, member of number of prestigious associations and academic bodies like International Society for History of Science USA, New York Academy of Science USA, British Society for Philosophy of Science UK. I now most humbly invite Father Joe to share his thoughts and expertise on developments in genetics and emergence of superhumans. I request the participants to post their questions in the chat box, which we'll take afterwards. And please write your name and affiliation. We would love to know from where all the participants are joining in. Father Joe, please.
Yes. Minute. Okay. First of all, thank you, Professor Naqvi, for your introduction, gracious introduction. And I consider it a privilege for me to address the participants of this webinar organized by SFS College, St. Francis de Sales College, Nagpur. As Dr. Susan already mentioned, this college is an outstanding academic institution in Central Asia. Before I proceed, can you people hear me properly? Can you hear
Yes,
me
Father.
and see me?
Yes, Father.
Thank you. Thank you very much. Thank you very much. Asst College and Indian Institute of Science, Delhi, have been collaborating from 2006 onwards, right from the beginning of CDRES. We have been collaborating, and it has been a very fruitful collaboration. And not only Dr. Susan and other people, also the principals, but particularly Professor Dr. Thomas, who is the principal today. He has been a very great supporter and has taken pains to encourage us, to support us, and take us forward. So I want to congratulate the organizers, Asst College organizers of this webinar, because they are the main organizers. Dr. Thomas, who is the principal, and Dr. Susan, and Praveen, who is helping, and many other professors of Asst College and well-wishers of Asst College. All these people have contributed towards setting up, organizing this webinar. I want to thank them, I want to congratulate them, and I want to wish them the very best, because this collaboration and this service to the college and to humanity must continue. And this is my wish and this is my prayer. Once again, I want to thank them for giving me this opportunity to share some of my ideas and thoughts with you all. And also I want to thank all of you who have taken pains during these pandemic times to participate in this. I noticed more than 100 have already signed in, and more are coming in. And so it's very encouraging to see so many people taking interest in this kind of programs. So welcome to all of you, and thanks to all the organizers. Let me now move into my topic. I have chosen a topic which I hope will be easier for you to understand, because not all of you are specialists in genetics. And also, I hope it will be profitable for you, not only for today, for the rest of your life. Okay, the theme I have is developments in genetics and the emergence of superhumans. As His Grace Archbishop of Nagpur already mentioned, this area of genetics is a very, very significant and very, very consequential area. So many new things are coming out, new things which will have a tremendous bearing on us today as well as tomorrow, future generations as well as present generations. And so we cannot be passive spectators in this development of genetics. And so many technological breakthroughs this development is bringing about. We have to understand this thing. We have to respond to these things in a responsible way. And that is why we organize this kind of seminars. Now, as I will point out to you that this development has got many, many consequences. I have chosen only one specific consequence. That is the emergence of superhumans. There are many others. The Archbishop, His Grace Archbishop, already pointed out to you that genetic developments are helping us with the food. The food production has taken a tremendous developmental process because of this. And also animal husbandry And various places, genetic revolution or genetic developments have impacted, and many new things have come, and we humans, we citizens of the world, are the beneficiaries of these developments. Anyway, that is by way of introduction. So let me just give a short summary of what I am going to do so that you will know where we are going. Science, particularly genetics, today has made incredible progress. Although many amazing developments are taking place in this field, in this presentation, I focus on the possibility of science helping to create superhumans with the special capabilities and talents. Obviously, this possibility is full of promises, positive things, and also perils, challenges, dangers. So we have to tread the path very carefully. Okay, I suggest that instead of opposing this development in science because of the negative impact it can cause, we can allow and encourage it for the good of all of us. But how can we do that? But to achieve this noble goal of the betterment of humans and the world around, a healthy, creative, and constructive interaction and integration between scientific ideas and techniques on the one hand, and sound, ethical, religious principles on the other. This is necessary. Science alone, if we allow, there will be serious problems. Religion alone, if we allow, there will be also serious problems. But we need to integrate these two, as the bishop, Archbishop mentioned to us. We need to find ways of maximizing the beneficial effects and minimizing the ill effects of these scientific developments. And how can we do this? We can do this if we allow genuine scientific developments to interact and integrate with genuine religious, ethical values. I will point out how this can be done, what are some of the criteria we need to use to carry out this happy balancing of scientific ideas and ethical principles. We will see that towards the end of my presentation. So let me now, after giving you the summary view of my presentation, let me go into some specifics. First of all, developments in genetics. What are some of the developments? And even before that, you may want to know, some of you may not be familiar with genetics, so you may want to know something about genetics. I'll be very brief because Dr. Susan and various others also will be talking about this. So I will not take too much of my time, and also I don't have much time to spend. So I will be very brief. So please pay attention as much as you can. What is genetics? Genetics is the study of genes and related issues. Gene is the basic unit of heredity. And the gene is made up of a succession of nucleotides. They are big compounds, big compounds of cells, or compounds of atoms and molecules and so on. In the human genome, there are about 3 billion nucleotide pairs. And so if you go by the number, more than 6 billion nucleotides. To be exact, I think 6.2 billion nucleotides. Now, who began all these things? You'll be surprised, some of you may not know. This whole business of genetics is not very old. It began by Gregor Mendel, a Catholic monk from Austria. Normally, people would not expect a Catholic monk to be engaged in this, but he did. He did. He's called the father of genetics. In 1866, for the first time, he identified gene And that was the beginning of genetics. We should be grateful to him. Very, very hard work he put in. He did not have the modern equipments. He had to do it with his hand and with his mind, and take time and time to count so many thousands and thousands of 8,000 plus P's he had to investigate and do. If you read his biography, you'll find tremendous contribution, tremendous work. Let us be grateful to him. Okay, let's proceed. Genome gives a clear idea of the genetic structure. So what is a gene? Genome is, the sum total of the genetic structure of our species. And human genome study tells us that genetically humans differ from chimpanzees only by less than 1%. The genome of chimpanzees, genome of us, humans, Homo sapiens, the difference between two of us is only less than 1%. So if with a little bit of change, we people who are attending this webinar probably would have been on top of trees and hopping around and doing things like chimpanzees. Thank God we have been privileged, and we are sitting in the classrooms or in our study rooms and listening to this. Chimpanzees are 99% humans genetically. Domestic cats, 90% humans. Cows, 80% humans. So you might say, oh, these are only animals. What about plants? Well, the banana which we ate, some of you may have eaten banana this today. 60% bananas share the genetic structure with you and me. We are all brothers and sisters. The whole living creation is brothers and sisters. We can really call ourselves brothers and sisters. We need to respect, even the banana we need to respect. We need to respect animals. We need to respect everyone. Science is telling us this. Science is revealing this truth to us. Okay, let's proceed. Some new developments in genetics. Human Genome Project, genome sequencing. I will not say much about it because of lack of time. Its goal was to determine the base pairs that make up human DNA, identify and map all of the genes of the human genome from both a physical and functional standpoint. And this was done in 2003, not long ago, and it was a tremendous breakthrough, incredible breakthrough. It has made so much difference to humanity. I don't have time to talk about it, especially in the medical field. The impact of this discovery, that is Human Genome Project or genome sequencing, is tremendous. Medical field. It is slowly being realized. These ideas are being applied to medicine slowly. But it is going on, and tremendous benefits we can expect from this. Of course, there will be some dangers. We'll talk about how to deal with the dangers later on. Let's proceed. Genetic engineering. Another development in the field of genetics is genetic engineering. In simple language, genetic engineering is basically engineering at the level of genes. You could say at the level of molecules. You could even say at the nano level. And this field has been going on for quite some time, and it has made tremendous progress and is still making tremendous progress. Let's hope that more and more new things come thanks to genetic engineering. Next one, cloning, another development. I'm giving you some general ideas of the various developments in genetics. Cloning. What is cloning? Cloning enables science to make exact genetic copies of any living being, including humans. In 1996, Dolly, you all have heard about Dolly, a domestic sheep was cloned by Ian Wilmot and his team in Scotland, in Great Britain. Since then, cloning was done on many other living beings. Practically all the important living beings have been cloned. Okay, I'll talk about, I'll mention or just briefly talk about human cloning later on. Okay, stem cell research, another area of very, very significance, great significance in genetics is stem cell research. And what are these stem cells and why are they important? Stem cells are, technical term, pluripotent cells. That is, they can grow into different types of cells. You know, in our human body, we have got different types of cells. Blood cells are there, bone cells are there, muscle cells are there, various cells. And, pluripotent means when they are in their initial stages, they're able to grow into different cells. But once they are, ordinary cells, once they are mature, become adulthood, they can grow only into that type of cells, not any other cells. Whereas stem cells can grow into various different type of cells. And this is a great blessing because if we have good stem cells, we can develop cells for making, creating a heart, or for making a liver, or making a bone, all kinds of things we can do if you've got stem cells. And that is why it is very useful for different therapies, medical therapies. And it is being used, in a limited way in various parts of the world. Okay. Genetic editing. This is very important. This has become extremely important in recent days. And Dr. Susan, the next presentation, will be talking about in detail. So I will be just giving you some basic ideas to be in this presentation. The DNA of life is deoxyribonucleic acid. DNA is an acid, nucleic acid. It naturally stores its information in just 4 key chemicals: guanine, cytosine, adenine, and thymine. And they are usually referred to in their first letters, G, C, A, T, respectively. Now, human life can be considered a book of life. And how is this book of life written, or in what letters, alphabets is the book of life written? It is written in 4 letters, the same nuclear chemicals, key chemicals, G, C, A, T. Hence, like a book, DNA can be edited, modified. This process of modifying, editing DNA is called genetic editing. And this has got tremendous significance, as Dr. Susan and others will explain to you later on. Let me proceed. CRISPR, another great discovery, just recent times only, and this got a Nobel Prize. The person who contributed substantially to this was given the Nobel Prize this year. So CRISPR, this year means just 2020. I mean the CRISPR, and its long name, clustered regularly interposed short palindromic repeats, is a very simple, inexpensive, but very effective method of gene editing. You will see that in the next presentation. So I will not spend any more time on this. Just want to mention this is one of the most important developments in genetics recently, and it has got tremendous impact. And this has revolutionized, CRISPR has revolutionized genetic editing, genetic modification. And that is why the discoverer or the inventor of CRISPR was given a Nobel Prize just recently. Let's proceed. Okay, now, so I have given you a bird's eye view of some of the important developments that have taken place in genetics. Only some of them. There are many, many more. Okay. And now I want to see how these things are going to impact on us humans. How are we going to be affected by this, positively or negatively? And as I mentioned to you, I'm only considering one specific case because of lack of time and various other restrictions. Possibility of emergence of superhumans. Superhumans, producing superhumans is within the control of science today, within the purview of science today. And superhumans would mean people with special abilities. Originally my plan was to tell you that this kind of developments can give rise to a new species, post Homo sapiens species, or trans Homo sapiens species. And I thought before we go into new species, let me talk about something similar, that is superhumans, great heroes, especially gifted people like Einstein and various other people. Possibility of emergence of superhumans because of genetic developments. And why do I say this? Some favorable conditions. There are certain conditions which genetics have revealed to us, which encourage us to think that production or emergence of superhumans is not just a theoretical possibility, but it's a practical probability. Why? What are some of the things? Let me mention here. Genetic difference between two close species is very small. I already mentioned to you between chimpanzees, and there are other beings also, bonobos or something like that. Between these beings and humans, the difference is only 1% or less than 1%. Hence, by tinkering with the DNA of a chimpanzee or in the right way, we can create a human being. By tinkering with the DNA of a human being, ordinary human being, we may be able to create a new species, a superhuman species. Okay. As I say, we may not go that far about making a new species, but at least superhumans, special humans with the special abilities, we may be able to produce it, because genetically the problem is serious, but it's only 1% or less than 1%. And science today is capable of making that kind of changes in the genetic structure because of Human Genome Project and genetic engineering and so on. 1% of change in the right way, to make in the right way, is within the power of science today. Okay, and that is one encouraging thing for me to believe that emergence of superhumans is possible. Next, the Chinese gene-edited twin girls. You may have heard about this. I have read about it, I'm sure. In 2018, Chinese Dr. He Jiankui made the first genome-edited twin baby girls, Lulu and Nana. He successfully used CRISPR-Cas9 technique, and the girls were claimed to be HIV, AIDS-free. And CRISPR-Cas9, Dr. Susan will be, explaining this thing in detail, so I will not say anything about it. Using this technique, Dr. He Jiankui was able to produce gene-edited twin baby girls, and he did genetic engineering on their fetus, and as a result, these little girls were claimed to be HIV free. Their father had HIV. Mother did not have. Father had HIV. And so there was a possibility that these girls, or at least one of them, will contract HIV. Dr. Jiankui claims that they will not contract this one. The point simply is that today, tinkering with the genes in your womb, when you are in your mother's womb, is possible, and Dr. Jiankui has shown it. Of course, he had problem. He's still in jail. He's put in jail. What has he put in jail? At present, this kind of experiment on humans are legally banned. So he did not follow the legal restrictions. So he is, as far as I know, he's still in jail. But this instance shows that the method is successful. The method works. It works. And if you know a little bit of history of science, science always begins small. You know, today to travel to the US, you know, from Delhi, I'm speaking from Delhi to San Francisco, that's the longest air route in the world today. And we have our Indian ladies piloting it. Fantastic, piloting it. And it can be done non-stop, can be done. And this is today, air travel. But in 1912, when the Wilbur and the Wright brothers, Wilbur and the Orville brothers started space travel or the air travel, they went only a few meters. Some meters only they could travel. That is what happened in 1912. And today we can travel such long distances for heaven, and of course much more. So science always begins small. Jiankui may have started small, all right, but it can grow, it can expand, it can even explode. So the chance, the possibility, it is not just a theoretical possibility, it is I would say, a practical probability. We have to consider it very seriously. Okay, let's proceed. Another case, clonade and claims of human cloning. I mentioned about cloning earlier. Now, human cloning. Clonade founded, I will not go into detail. On December 27, 2002, Clonades is an association. Chief Executive Brigitte Boisier claimed that a baby clone named Eve was born. She announced this one, but people are very skeptical. I'm not so skeptical about it. Let me confess, I'm not skeptical about it, because according to me, science has the resources, science has the means to do this. Of course, the law does not allow. So she and her other people have kept it secret. Anyway, we do not know whether it is true or not, but this lady is making the claim. It has not been confirmed by the scientific community, but I personally think that the possibility or even the probability or even the reality cannot be ruled out. Okay, given all these things, there are only two instances I gave. I can give many more instances. I think that science is in a position to produce superhumans. Superhumans here, what I mean is humans with enhanced capabilities, humans with enhanced abilities, special abilities, which ordinary people will not have. And that is the beginning of a new species coming up. This is possible. Now, this is a highly complex and controversial issue. That is science creating superhumans. It's a highly controversial issue. However, there seems to be no in principle scientific serious objection to this. Why? Because the idea and reality of superhumans is not new. Here I'm doing a little bit of my own ideas. All religions take pride in their superhuman heroes. Think about Peter Damien of Molokai, a Christian priest who worked among the lepers of Molokai, and of course he became a leper himself and died. Think about Mother Teresa of India. These people are honored as superhumans in the world of service and spirituality. What do I mean by this? We talk about loving our brother, sister, and making sacrifice to them and so on. For us, for you and me, at least for me, it is a great ideal. I will say to Mother Teresa, I know, I respect you, I honor you, I all these things, but Mother, I'm sorry, I will not be able to follow you. I don't have that kind of spiritual strength, but these people have. These great saints and sages have. And I consider them as superhumans. Let us now proceed to other religion. Sri Aurobindo, in our Indian tradition, talks about Gnostic humans. He talks about evolution, spiritual evolution, and then he talks about Gnostic humans. And according to his thinking, we humans eventually become Gnostic humans, of course, provided we satisfy certain conditions. So there are, in the Indian tradition or Hindu tradition also this kind of thing. In Buddhism, there are bodhisattvas, who are enlightened persons who delay their attainment of salvation to help others attain salvation. These are great saints, bodhisattvas, in the Buddhist tradition. And we know our rishis and other sages had superhuman powers. Many of our rishis and munis and other sages, they had superhuman powers. We know that. We have, that tradition in us. Okay. But at the same time, when science attempts to make superhumans, the matter becomes highly complex, and so appropriate conditions will have to be put in place. And so I like to talk about some of the conditions that is required. Before I talk about the conditions under which science may attempt to make superhumans, certain conditions are to be satisfied. We are going to talk about that. Some remarks. We have seen that science today is in a position to make humans with enhanced capabilities. However, since the consequences of this venture are enormous, it has to be done under serious, stringent conditions. In my view, this is a paradigm example of science and ethical or religious values interacting in a creative way. And so some ethical values I'm going to present to you, which need to be taken seriously when science attempts to create superhumans. The first ethical value, I put it as public beneficence. These are values used by other agencies also. It is not my own creation. I accept it, but I got it from other sources. Public beneficence. What does it say? This principle demands that scientific action or involvement must make sure that the public benefits are maximized and the harm is minimized. Benefits maximized, harm minimized. This will put a break to all forms of greed for money profit. You see, you can have some, designer babies. Today, what I'm talking about also refers to designer babies. That was a topic 10 years ago. Nowadays, people don't talk much about designer babies. Science can design new type of babies if you have plenty of money and you give money to scientists. It is possible that scientists will design special babies with enhanced abilities. Okay. And a company can take that up, a private company can take it up and start doing that service for profit, for money, and they don't care what happens to the rest of humanity. No, you cannot do that. That's what this principle is saying. Scientifically possible is no guarantee for ethically permissible or humanly desirable. This is the principle. Scientifically possible does not mean it is ethically permissible, or it does not mean that it is humanly desirable. No, you have to take this principle seriously into account. Next one, responsible stewardship. What does this mean? This principle calls for concern for those who are not in a position to represent themselves, such as children and future generations. You know, you can, you say, we got the technology, we got the techniques, and we got the money, so we are going to do it. Sorry, sir. You have to think about children. How will it affect children? Even children who are in the womb. And how it will affect future generations. You will come and go. When you die, you are finished. What about the future generations? You cannot put something here which will harm future generations. No, you have to take this thing into consideration very seriously. This principle stipulates sensitivity to the well-being of the poor, uneducated, and the voiceless. No, we cannot simply ignore the voiceless. We cannot ignore the uneducated people. Oh, they don't do what they want. I am going and get my child if the special abilities, and so that my child will score the highest in IAS exams and other things and so on. No. You cannot simply do like that. You also have to think about how your action is going to affect other people. We are all one community. We are all brothers and sisters. Okay, we are all members of a family, and so we have responsibility for our brothers and sisters. We cannot ignore them. We need their help because we are not islands. Because we don't live alone. We need the help of our brothers and sisters, and we need their help. Then we should be also sensitive to their needs, to their well-being, and other things. Okay, next one, intellectual freedom and responsibility. This principle refers to moral accountability of the people responsible for the innovation. Scientists cannot say, I'm a scientist, I do science, so I don't care what happens to other people. No, sir. You have a moral accountability to the members of society. Okay, intellectual freedom is necessary for any creative work, but certain limits and restraints will have to be put in practice so that harmful effects will not take place, or at least will be minimized. So that is what this principle is saying. This ethical principle is saying. Last one I'm mentioning. There are many more. Democratic deliberation. This principle stipulates that serious decisions in the context of any innovation have to be done in a collaborative way. You know, you have to engage in serious discussions and debates, and you have to take into account the opposing views and active participation of all stakeholders. You see, you have to, before you venture into making superhumans, you have to see the pros and cons, and you have to listen to people who may not agree with you. Not because you don't like them, no, but you want to find out what some of the problems are. And it has to be done in a collaborative way, where different people will be able to give us different ideas. We take all these ideas into account, and then we proceed. For instance, let me just say an example. Science tells us that efficiency is very important. Success is very important. Okay, you can, efficiency is very important. By all means, have efficiency. And by all means, have success. But also remember, it's important, compassion and concern for your brothers and sisters is also important. So your efficiency and success will have to be balanced with compassion and concern for other people. And efficiency and success will come from science, compassion, and concern for the voiceless will come from religion or from ethics. Both will have to be taken. Then only you have got a happy society. Then only we have got a society where everyone feels happy. If you got a society where happy people are happy, happy people are not happy, it will not be a pleasant existence. There'll be always problem. Nobody will gain, nobody will enjoy life. No, we want to make sure that everyone gains, everyone enjoys life through science. And for that, I'm saying these principles, these ethical principles, and many more, number of others. According to circumstances, we have to add more principles will have to be done. And so I'm coming to the concluding remark because my time is almost over. Let me just give concluding remarks and finish it. Recent developments in genetics are very resourceful and present before us very valuable possibilities. But they are also ridden with serious risks. It seems to me that the response to this critical situation should be constructive, creative, and not confrontational and condemnatory. In my view, rather than pressing the stop button on these scientific achievements, the most appropriate strategy will be to guide these developments along the right channels so that humans and our universe are placed in a better, more enriched, and nobler trajectory. And to achieve this most challenging task, sound and time-tested ethical principles will have to be integrated into scientific ideas and technological know-how. All these render science ethics or religion interfacing or interaction as a necessary step in our world today. Indeed, thanks to genetic revolution and various other revolutions, we humans are on the throes of a fast advancing scientific and technological revolution. It may not be an exaggeration to say that in our scientifically fast advancing world, science ethics or science religion dialogue is not just an option, but is becoming an obligation. We cannot ignore this. We can ignore this only at the peril of bringing serious harm to our society. So we need to go forward. We need to encourage science ethic interfacing dialogue, and that is what CDRES, IASR, and other such organizations are doing, not only in India, worldwide. Let us wish all of them success so that our human family grows, enriches itself with science, with genuine religious ideas or ethical principles, so that we'll have a happy community, a richer community, and a nobler community. This is my wish and my prayer, and I am sure you also have the same sentiment. Thank you very much for listening to me. I'm done. I'm done, sir.
Thank you, Father Job. Thank you, Father Job, for your enlightening and thought-provoking address on a very debatable and a very interesting topic. We are indeed blessed to have you as today's guest speaker. You have thrown light on various aspects of genetics like Human Genome Project, genetic engineering, cloning, stem cell research, gene editing, and a very recent thing, CRISPR. You have rightly pointed out that possibility of creating superhumans is filled with promises and perils. And I fully agree with you, Father, that healthy, creative, and constructive interaction, integration between scientific ideas, technology, and sound ethical principles is of utmost importance. Your talk has invoked a lot of interest among our participants. From this, I can make out from the wonderful comments coming in the chat box. Father, with your kind permission, may I take up a few questions by our participants?
I leave it to the organizers. I have taken my full time. So if you allow me time, I will be happy.
Father, we would love to interact with you on behalf of our participants. Okay,
sure.
Just quick questions. Okay. Father, there's a question by Dr. Jessica Samuel. How can we maintain the uniqueness of humanity as being created in God's image and likeness by application of genetic engineering?
Yes, first of all, when you say that we have been created in the image and likeness of humans, what do we mean by that? That it can take many different types of meanings. And God has given us many talents. And developing these talents and fulfilling God's design, God's plan for us is part of our mission. And so, how can we do this? I would simply say, give the answer, as I gave in my concluding remarks. We make use of the talents God has given us. That is, that is part of being in the image of God, because God has got all those talents with God in an infinite way. So make use of these talents, but develop them. And for developing this talent, science can help us. So use science as a means for developing it. And when you're developing it, as I already mentioned, make sure that selfishness and self-centeredness will not be the criterion. The criterion which we have seen, some of the criterion we have seen, will have to be used. And this person, as I would say, is quoting from the Bible. So the Bible itself gives a lot of criteria for developing our talents, so that we will become more and more like God. Our imaginess of God increases. So I would say use science, but use science in a responsible way, as mentioned in this talk.
Okay, thank you, Father. Father, I would like to now take up question posted by Dr. Samira Siddiqui. How the stem cells from plant origin can be beneficial to the humans, as lot of such patented products are available in the market which claim to have remedial properties.
Yes, this stem cells, as I mentioned, stem cells are cells, with very special type of cells, which can grow into various different cells in our human body. And how can they help? They can help in various ways, depending on what your need is. So you can arrange the development of the stem cells to go along that direction, so that that particular type of cell will be produced in the adult stage, and then make use of that cell for whatever work you want to do. That is the advantage of stem cell. For instance, you're talking about plants. Plants, the plant may have a particular sickness, and because of that sickness, that plant may be deficient now, and it may have lost a certain special type of cells. So by stem cell therapy, you can provide that plant with these cells, and then that plant will be revived. That plant will be able to function normally. So it's a thought of a therapy. Normally we talk about therapy for humans or animals, not so much for plants, but for plants also it can be done, because the principle is the same.
Okay, thank you, Father. Father, there is another very interesting question which I would like to take up from the participants. Do you think superhumans will have edge over normal human beings in different parameters? And what type of society do you foresee if there are superhumans along with the normal human beings?
Yes, that is a very, very interesting and very serious question. Okay, and this is going to create problem. I have done some thinking about it. Now I mentioned, that's why I took the topic as superhumans rather than a new species. Transformer sapiens species will create big problems. Okay, now superhumans, say for instance, if the superhuman beings are really following the ethical principles mentioned here and many others, depending on the circumstances, then there will not be any problem. For instance, think about, I suppose the person who posed the question will accept that a person like, some of our rishis. Let's talk about some of our rishis are superhuman beings. They have got superhuman abilities, special abilities, which normal people don't have. But they are using their power for the good of us, for good of good of us. You go in our scriptures or in our tradition. We know people going to rishis and getting help, advice sometimes, or certain techniques that they teach you, yoga and other type of techniques they teach you, and they are helping us. So having these superhumans in itself need not be a disadvantage to community. Only thing is, if the superhumans are the rishis and so on become selfish and self-centered and they don't care about others, they only say, I got the advantage, I got this power, that power, so I can have advantage, I can make plenty of money, I will ignore it. If they take that kind of stand, there will be a problem. And hopefully great people will not take that kind of problem. Will create that kind of problem. So there will be problems, and that is why I'm saying ethical principles will have to be applied, not only in the creation of these superhumans, but also in their behavior. We have to tell them that, okay, you have got these special abilities. That does not mean that you can become multi-trillionaire and we all have to starve. No, you have to share what you have with us. We have to tell that person. So we'll have to have ethical principles or religious principles, as and when required, and apply them. And that, I think, will make sure that these superhumans become a blessing for us rather than a curse.
Yeah, very true. I think this is something akin to Father artificial intelligence, which is coming up.
Yes,
are using it.
I did not, because of lack of half an hour too short, because of half of this, I will not go into artificial intelligence. That also part of the enhancement of human abilities.
Yes.
Yes, cyborg. Now they are now thinking about actually working on. I have read of those things. They are working on a human-machine interfacing. For certain things in your brain, you put a super computer or something like that, so you can do certain things very fast and in a very efficient way. That is called sort of a cyborg, and that is man-machine. They call it human and or man-machine interfacing, and that's going to come. Already they are experimenting on that.
Yeah, very true. I would like to add one thing. I think this superhuman creation is like a double-edged sword.
Absolutely.
We have to treat the path cautiously. And there has to be a line. We have to draw a line. What is ethical and what is not ethical.
Absolutely.
Yes, Father. So that was wonderful interacting with you, Father, on behalf of the participants. And thank you so much, Father, for guiding us and highlighting a very important topic, which is a controversial and obviously a debatable topic, and making us aware on the ethical aspects of genetic engineering and cloning. On behalf of the management and principal, I express my sincere gratitude to our guest speaker of today's session, wonderful session, Father Joe, for his enlightening talk and touching upon a very, very interesting topic. Thank you so much, Father. Thank you for connecting with us.
Thank you for the opportunity. Thank you very much for the opportunity. Thank you. Bye.
I now hand over the proceedings to Brother Daniel. Over to Brother Daniel.
Hello.
Yes, brother, you can continue.
Yeah, am I good to go?
Yeah.
Okay, thank you, Dr. Nikhat, ma'am. Good morning and a warm welcome to all to the second session of this national webinar. I, Daniel D'Souza of BSc third year, SFS College, am glad to introduce to you the speaker of the second session, Dr. Susan John. Dr. Susan John is an assistant professor of physics at St. Francis de Sales College, Nagpur. She has a teaching experience of 15 years at UG level and 2 years at PG level. She is also the coordinator of CDRIS at SFS College, which is organizing this webinar and also co-convener of today's event. She has many awards to her credit. To name a few: award for scoring highest marks in RTMNU MSc Physics, which includes PL Kare Award, RCK Chandurkar Memorial Gold Medal, KBL SAT Gold Medal, and she is also one of the recipients of award for 13th National Seminar on Crystallography. She has presented and published many papers in the field of physics and science-religion dialogue. A hearty welcome to you, dear ma'am. We are eager to listen to you. I now kindly request you to take over the proceedings. Thank you. Over to you, ma'am.
Suzanne ma'am, unmute yourself. Suzanne ma'am, unmute yourself.
Hello, I am audible. Hello.
Yes, ma'am. You
Hello.
are audible.
Actually,
Yes,
I'm
yes.
facing
Yeah, ma'am,
some
you are
problem
audible.
with the mic. Please wait for a second. Can you tell me where is the share screen? Hello. And that, wait for a second, please. I'm very sorry. Can you see my screen?
Yes, ma'am.
Yes, ma'am. It
Yes,
is visible.
yes.
Yes, ma'am.
You can make it bigger. Okay, yes, sure. We can see it. We can see you.
Yes, Father. Yes, Father. Good morning. I'm really very sorry. Some technical issues occurred. So my topic is recent developments in genetics and its ethical responsibility. So we are going to know what is genetics, and then we will see the ethical responsibility when the researches are going on, then the scientists should take up. So what is genetic engineering? Genetic engineering or genetic modification is the direct manipulation of an organism's genes using biotechnology. It is a set of technologies used to change the genetic makeup of cells, including the transfer of genes within and across species boundaries to produce improved or novel organisms. New DNA is obtained by either isolating or copying the genetic material of interest using recombinant DNA methods or by artificially synthesizing the DNA. Then, what is DNA? Then how DNA is converted to RNA and last it to the protein. So DNA, deoxyribonucleic acid, is a macromolecule composed of two polynucleotide chains that coil around each other to form a double helix carrying genetic instructions for the development, functioning, growth, and reproduction of all known organisms and many viruses. The two DNA strands are known as polynucleotides as they are composed of similar monomeric units called nucleotides. The nucleotides are joined to one another in a chain by covalent bonds. Both strands of double-stranded DNA store the same biological information. This information is replicated as and when the two strands separate. The two strands of DNA run in opposite directions to each other and are thus anti-parallel. RNA strands are created using DNA strands as a template in a process called transcription, where DNA bases are exchanged for their corresponding bases, except in the case of thymine, for which RNA substitute uracil. Under the genetic code, these RNA strands the sequence of amino acid within proteins in a process called translation. Within eukaryotic cells, DNA is organized into long structures called chromosomes. Before typical cell division, these chromosomes are duplicated in the process of DNA replication, providing a complete set of chromosomes for each daughter cell. In eukaryotes, DNA is located in the cell nucleus with small amount in mitochondria and chloroplast. In prokaryotes, the DNA is held within an irregularly shaped body in the cytoplasm called the nucleoid. The genetic information in a genome is held within genes, and the complete set of this information in an organism is called its genotype. A gene is a unit of heredity and is a segment of DNA that influence a particular characteristics in an organism. In many species, only a small fraction of the total sequence of the genome encodes protein. In all organisms, two steps are required to read the information encoded in a gene's DNA and produce the protein it specifies. First, the gene's DNA is transcribed to messenger RNA inside the nucleus of the cell. Then messenger RNA is translated to protein in the cytoplasm of the cell. The transmission of genes to an organism's offspring is the basis of the inheritance of the phenotypic trait. The genes makes up different DNA sequence called genotypes. Genotypes, along with environmental and developmentals, determine the nature of phenotypes. Now we will see tools which are used to change the genome of the organism as follows. Restriction enzyme, the original genome editor. Zinc finger nucleases, increased recognition potential. Talon gene editor, single nucleotide resolution. Although all the above tools have significantly improved the capacity for precision, recently yet suffer serious limitations. These limitations are overcome by a new technique called CRISPR-Cas9. What is CRISPR? CRISPR means clustered, regularly interspaced, short palindromic repeats. Is a family of DNA sequence found in the genome of prokaryotic organisms such as bacteria and archaea. Cas9 or CRISPR-associated protein 9 is an enzyme that uses CRISPR technology as a guide to recognize and cleave specific strands of DNA that are complementary to the CRISPR sequence. Means this protein Cas9 acts like a pair of molecular scissors capable of cutting strands of DNA. Latest 3 CRISPR technologies are being used. First is CRISPR-Cas9. Second is CRISPR-Cas12a, formerly CPF1. CRISPR-Cas13, formerly C2C2. Again, I would like to throw some light on CRISPR-Cas9. For CRISPR involved technology, first is Cas9. CRISPR-Cas9 in its original form is a homing device in which the CRISPR part that guides molecular scissor, the Cas9 enzyme, to a target section of DNA. They together disables or repair a gene or insert something new where the Cas9 scissor has made some cut. CRISPR-Cas9 edit genes by precisely cutting DNA and then letting natural DNA to repair itself. The system consists of two parts, Cas9 enzyme and a guide RNA. Cas9 CRISPR-associated protein 9, formerly called Cas5 CSN1 or CSX12, is a 160 kilodalton protein which plays vital role in the immunological defense of certain bacteria against DNA viruses and plasmids. The most popular CRISPR-Cas9 originating from Streptococcus pyogenes SP-Cas9 belongs to the type 2 CRISPR system. Second technology, Cas12a, formerly CPF1. According to Zecte and co-worker in the year 2015, found CRISPR nucleases in Prevotella and Francisella 1 bacteria termed Cas12a CPF1 belonging to the class 2 type 5 CRISPR system. Additional effectors protein of this family include Cas12b C2C1 and Cas12c C2C3 respectively. Chamkov and co-worker in the year 2015 found Cas12a is an endonuclease which varies in size between 1200 and 1500 amino acids. And to note that Cas12a is more precise and more accurate than Cas9 technology according to recent studies. Cas13, formerly C2C2, the CRISPR-Cas13 family of enzymes target and cut RNA instead of DNA. Hence, this technology is used to kill directly RNA viruses. Let us see the history of genetically modified organisms. The first genetically modified organism was bacterium, made by Hilbert Boyer and Stanley Cohen in the year 1973. In 1974, the same technique was applied to mice by Rudolf Janisch. In 1994, the first genetically modified food were made available, that is genetically modified tomato, also called as flavor saver tomato, and its chemical name is CGN 89564-2. What is genetically modified crops? According to the research papers published by Milancevic and co-worker in the year 2008, genetically modified crops have been developed in an attempt to improve food quality and solve some of the problems associated with commercial agricultural, including disease and weed management. Let us see few examples. First is tomato, flavor saver, as I already told, also known as CGN 89564-2, a genetically modified tomato. As we all know, tomato have a short shelf life and it is more susceptible to being damaged by fungal infection if broken. And ripe tomatoes get easily damaged during the transit from growing areas to the market. And also climate of the region affects it. Hence, in order to address this problem, tomatoes are picked while they are green or unripe, and when it reaches the market, it is ripened by passing through ethylene gas, which act as a plant hormone. That is, the tomato does not complete its natural ripening process, which beta polygalactoronase. This research was unsuccessful as the anti-sensed PG gene had a positive effect on the vitamin A deficiency. If not treated, has numerous negative health effects such as dryness of the eyes, reduced immune system response, etc. In the developing countries, nearly Every year, 7 lakh children die and nearly 5 lakhs suffer serious childhood blindness due to deficiency of vitamin A. As rice is a staple food crop for over half of the world's population, a variety of rice produced through genetic engineering called golden rice, in which beta-carotene, a precursor of vitamin A, was induced. Now, let us see Bt cotton, the famous Bt cotton. Cotton gets easily infected by pests as they can destroy it fully. Hence, a genetically modified pest-resistant cotton plant, commonly called as Bt cotton, was produced. As per data available, in 2011, India grew the largest genetically modified cotton crop, followed by USA and China. According to data from Union Ministry of Agriculture, the adoption of Bt cotton in India has increased the production of cotton to 34.89 million bales in 2017-18 from 8.62 million bales in 2002 and 2003. Introduction of Bt cotton reduced the use of insecticide and also reduced the cost of cultivation. As we all like brinjal, there is one genetically modified Bt brinjal is also available. The genetically modified brinjal is a transgenic brinjal, also known as an eggplant, created by inserting a crystal protein gene CRY1AC from the soil bacterium Bacillus thuringiensis into the genome of various brinjal cultivars. The insertion of the gene along with other genetic elements such as promoter, terminators, and an antibiotic resistance marker gene into the brinjal plant is accomplished using agrobacterium mediated genetic transformation. The Bt brinjal has been developed to give resistance against Lepidopteran insect. I will not give much example. This is my last example, Bt banana, genetically modified banana. India is one of the largest producers of banana. According to survey report, it is the 4th most important food crop after wheat, rice, and corn in terms of gross value of production. Scientists have used latest gene editing techniques to modify the banana genome. The use of gene editing technique CRISPR-Cas9 in banana could serve as a useful tool that can be deployed for edible vaccines, improving nutritional quality, agronomical important trait, as well as pathogen resistance in banana. Many similar products are also coming up. First is herbicide tolerant soybean, insect resistant corn, virus resistant plum, vitamin enriched rice, vaccine from tobacco for hepatitis B virus, oral vaccine from maize. Many studies were conducted to find the effect of genetically engineered crop about the safety of GM crops with respect to possible long-term adverse effects on the environment, human health, and also on the sustainability of this new agricultural technology. According to the paper published by Prem and Lambert 2003, Precost and co-worker in the year 2005, GMOs have been associated with allergic reactions because the inserted genes usually come from other organisms such as bacteria. The proteins they produce are often new to animal or human diet. The production of protein may also involve a new biochemical genetically modified crop, a potential risk for sustainable agricultural pathway in the plants or affect an existing one. As explained by Father, cloning also comes under this section, cloning was done using CRISPR-9 technology. I will not go much into the details because of the lack of time. So now I am moving towards researches in genetics and its ethical responsibility. First is to ensure good health. High quality research is essential to achieve global health. Developments in genetics should ensure good health and well-being of all.
Hello?
Hello, Father.
Yes.
Second, preserve biodiversity. The life of the ecosystem is designed in such a way that species depend on one another for survival by means of food chain. Food chain refer to the sequence of transfer of not only matter but also energy in the form of food from one organism to other. It starts from producers such as grass and ending with a decomposer organism such as fungi or bacteria. The natural interconnection between food chains makes it food web. If any of the species dies off, it can set the entire food chain get imbalances. If genetically modified plant or animal came into existence, then it should not endanger any of the existing species. For example, if because of genetic modification, production of pollen get affected, then it will affect bees and ultimately entire ecosystem. Another issue concerns the spreading or crossing of genes from genetically modified crops. The animals that eat genetically modified crops will be affected, may have more tolerance to pesticide or insecticide, and this may harm the species and ultimately biodiversity too, according to the study conducted by Maclean 2005. Genetic modification should not have devastating impact on biodiversity. Vaccines. Now, world is facing a serious deadly disease called COVID-19. Coronavirus is an infectious disease caused by a newly discovered virus. First, one should understand how vaccines work. As I already told, DNA creates RNA. This mRNA acts as messenger. This messenger RNA just carries instruction from the assembly of proteins from DNA template to the ribosomes by producing proteins. This technology are being used to create vaccine. Hence, by this method, coronavirus can be stopped from entering human cells by producing spike proteins. Hence, if genetics can reconstruct specific sequence that encode for protein, then it will be a great help to human beings. But at the same time, vaccine should work better for patients, treat illness to improve the number of people who are survived, and also it should improve the quality of life for people suffering from illness. Genetic modifications should find some vaccine which will prevent recurrence of disease and reduce the number of people who become ill. Manage climate change. Climate change can have devastating consequences for biodiversity. By reducing carbon footprints, we can reduce the impact if genetic modification leads to replacement of fuels such as petrol and diesel, which will emit less greenhouse gases. That will be a great boon, but without affecting the biodiversity. For example, if freshwater bodies are essential to biodiversity, if newly developed genetically modified crop varieties that use less water, it will found a great importance. It should ensure less pollution. As we know, pollution refers to the very bad condition of environment in terms of quality and quantity. The rate with which the air pollution is increasing, not only in the country but also in the world, is alarming. The major sources of air pollution are burning of fossil fuels such as coal, oil, etc. Vehicle pollution such as gasoline, industrial pollution. Any researches in genetic modification should ensure that it should control the damage caused by air pollution. Thus, finding some substitute for fossil fuels, it will be a great help. Agricultural requirement. In order to increase productivity, agricultural activities make use of pesticides and fertilizers. Agricultural practices are using pesticides on a large scale because it reduces the growth of unwanted weeds, disease, insects, and pests. These pesticides and fertilizers are the main source to contaminate the surrounding air. Nowadays, pesticides and fertilizers are mixed with new chemicals for quick growth of the crops and vegetation. Once they are sprayed over, the smell and the effect of the pesticides are left in the air. Some gets mixed with water and some slips into the ground, which not only destroy the crop but also uses numerous health related issues. As per data, the death due to chronic disease by overuse of pesticide poisoning is increasing day by day. Actually, it is affecting every form of life from fishes to birds and also to the human. Exposure to pesticide has adverse effects to humans such as stinging eyes, nausea, diarrhea, cancer, etc. India is one of the worstly affected countries. With the help of genetic modification, farmers can grow more crops on the same amount of land with less or no use of pesticide, but it should conserve biodiversity. Conclusions. Biodiversity provides humans with raw materials for consumption as well as production. Biodiversity not only provides livelihood to humans, but because of its proper functioning supplies pure and clean air, oxygen, water, etc. Any deterioration in the condition of biodiversity can affect human beings. There is an integral connection between human and biodiversity. Whatever modifications are taking place, it should not interfere the food chain, that is biodiversity. It should not create long-term adverse effect on the environment and human health. At the same time, it should not cause serious effect on biodiversity. With COVID-19, we have seen the damage the disease created not only to humans but also to the global economy. By protecting biodiversity in the Earth's ecosystem, we can prevent future pandemics. Thus, quality and ethics need to be embedded in all the areas of research of genetic modification. Hence, I can only say development and growth is essential, but that all should be done with morality and proper ethics. Thank you.
Thank you, ma'am. It was pleasure listening to you. You have brought home to us the basics of genetics, that of genetic engineering and the tools involved in it, and also shed light on our ethical responsibility and response towards it in a simple yet effective manner. Now, there are a few questions asked by the participants. With your permission, may I put them forth before you,
Yes.
ma'am?
Yes, yes.
First question, are GM food products, that is genetically modified food products, are safe and healthy?
Well, the answer is 50-50. As I told, take case of golden rice. It will help. Am I audible? Hello? Hello?
Yes, ma'am, you are.
As I told, take case of golden rice. It will help to eradicate the vitamin A deficiency. But there are research papers showing results that these genetically engineered food can cause the development of disease which are immune to antibiotic. Hence, my answer is genetically modified food has both positive and negative effects.
Okay, thank you, ma'am. And the second question would be, what is cloning and how it is, I mean, how does the CRISPR-9 technology is implemented in that?
CRISPR cloning. Okay. CRISPR cutting and ligation of nucleic acid in vitro was devised to enable efficient cut and paste of multiple complex DNA fragments by using CRISPR-Cas9 technology. First cloned animal was tadpole came into existence in the year 1950. Then in 1996, Dolly the sheep was successfully cloned. And whatever I told in my lecture, the same procedure is applied for cloning also. Thank you.
Yeah, ma'am. Thank you very much. I would like to express sincere thanks to you once again for sharing your valuable inputs. Thank you. Now, before we could call it a day, I would like to brief tomorrow's program. Tomorrow's first session, we have Dr. Firoz Hossain, a principal scientist, Maize Breeding Division of Genetics, IARI, New Delhi, who will be delivering a lecture on nutritional quality enrichment in maize through genetic avenues, followed by the second lecture by Dr. T. Srinivasu, Head, Retired, Department of Molecular Biology and Genetic Engineering, RTM Nagpur University, Nagpur. He will be delivering a talk on SARS-CoV-2 and its impact on religion. Then we have Dr. Victor Ferral, Dean of Philosophy, Rashol Patriarchal Seminary, Goa, who will be speaking on ethics of gene editing. And followed by Father Dr. Joe K., who will be concluding and summing up all the sessions, which will be followed by valedictory function, and we will end the webinar with the national anthem. I thank all the participants who have joined us today to listen to us on this webinar on genetic developments and their impact on human and society. Thank you very much, dear participants, on behalf of SFS College and the CEDRES team. Do join us tomorrow for more enlightening and enriching sessions. Take care. Thank you.